session is about to begin. May I request you to kindly mute your microphones and wait as we prepare for the session. Thank you. Ladies and gentlemen, our next session is about to begin. May I request you to kindly mute your microphones and wait as we prepare for the session. Thank you.
Good morning, everyone. Welcome to the RB Memorial Conference on Translational Research in Medicine, TRIM 2022. Welcome to Track 4 on Proteomics. Proteomics and omics technologies are making huge revolution in areas of biology and medicine. Mass spectrometry and various immunoassays based discovery of biomarkers that critically define the disease and the health state, along with their corresponding pathways, are currently being investigated for various diseases. Additionally, various new proteomic technologies are able to now provide answers and clues for various questions which were not addressed previously. In this track, we are going to have three eminent speakers: Professor Michelle Hill, Dr. Suman Thakur, and Dr. Mahesh Kulkarni. Let me thank all the speakers for accepting our invitation and to grace this occasion. Good luck for Track 4 Proteomics.
Ladies and gentlemen, our next session is about to begin. May I request you to kindly mute your microphones and wait as we prepare for the session. Thank you. Ladies and gentlemen, our next session is about to begin. May I request you to kindly mute your microphones and wait as we prepare for the session. Thank you.
Good morning, everyone. I am the moderator for this track. My name is Neha Gayatri. Uh, I have Divya Nair with me also. Good morning, Dr. Suman Tapa. Are you available online? Dr. Suman Thakur, are you available online? Uh, Professor Michelle Hill, are you available online? Dr. Suman Thakur. Yeah, it seems like the first speaker is not available. So we will go with Dr. Mahesh Kulkarni's talk first. It is a recorded talk. Due to some circumstances, he has not been available today. But if you have any queries, please do put them in the chat box, and we will convey it to him via email. While posting your question, please add your name and your email ID along with the question so that it can be forwarded properly. Thank you.
Good afternoon, ladies and gentlemen. It is a matter of great privilege to have with us Dr. Mahesh Jay Kulkarni. Dr. Kulkarni is a scientist at the Proteomics Facility, Division of Biochemical Sciences, CSIR, National Chemical Laboratory, India. Dr. Kulkarni's research interests lie in mass spectrometry, chemical modification of proteins, and chemical proteomics. His research activity involves developing mass spectrometric application for the proteomic research and understanding molecular mechanisms of biological processes using proteomic approaches. His team is currently working on proteomics of diabetic complications, wherein they are trying to elucidate the role of glycated proteins and advanced glycation end products in developing diabetic complications. This project's long-term goal is to identify a diagnostic marker for diabetic complications, identify drug targets, and develop intervention strategies. Additionally, they are working on the identification of off-target of drugs for repositioning in novel applications and understanding the molecular mechanism of their toxicity using combination of approaches involving chemical proteomics, two-dimensional electrophoresis, and mass spectrometry. Please join me in welcoming Dr. Mahesh Jay Kulkani.
Good morning, all. At the outset, I would like to thank Professor Sanjeeva for giving me this opportunity to speak in the conference dedicated to Professor Rinti Banerjee. I would like to start my talk with her quote, "Science and innovation will allow us to make a difference towards inclusive healthcare." What I got to know about Professor Rinti Banerjee is she was instrumental in setting up translational research in IIT Bombay. She was a very accomplished scientist in the area of healthcare research. She was also a member of a research council of CSR NCL. Unfortunately, we did not have any meeting in her presence. We miss her inputs and advice in carrying out our research at Biochemical Sciences Division for CSR NCL. With this brief note, I would like to start my talk on glycation of glucose-sensitive lysine residues, K36, K438, and K549 of albumin is associated with prediabetes. Our lab works on mainly this particular post-translation modification called glycation. This is quite prevalent in diabetic condition, wherein the elevated levels of glucose non-enzymatically react with the plasma proteins, especially albumin and other abundant proteins like hemoglobin undergo modification. The lysine and arginine residue of protein are modified by glucose, leading to formation of tryptase that gets transformed to amyloid product. Amyloid product which undergoes glycooxidation, leading to formation of reactive dicarbonyls such as 3-deoxyglucosone, methyl glycol. These reactive dicarbonyls are much more reactive than glucose, and they further react with the protein, forming advanced glycation end products. Modification of proteins by glycation leads to change in structure and function, as well as the modified proteins or the advanced glycation end products. They interact with the receptor RAGE. So this interaction leads to oxidative stress and activation of inflammatory pathways. The well-known diagnostic marker, that's glycated hemoglobin or HbA1c, is also a glycated protein. The levels of HbA1c in healthy control are below 5.7, and in prediabetes, the HbA1c levels are between 5.7 and 6.4, and in diabetic condition, the HbA1c levels are above 6.5. HbA1c is considered as gold standard marker for the assessment of glycemic status, as it provides glycemic status of preceding 3 months, and this is used for the for deciding the treatment and management of diabetes. Although it's a gold standard marker, there are certain limitations, especially in conditions like anemia, blood loss, iron deficiency. HbA1c is not an ideal marker. As well as currently used analytical techniques do not accurately quantify HbA1c. And HbA1c doesn't predict diabetic complication. In the literature, there are several reports that debate the usefulness of HbA1c, especially in predicting diabetic complications. So these are few examples. For example, this particular paper says that HbA1c alone is a poor indicator of cardiometabolic risk in the middle-aged subjects with prediabetes, but is suitable for type 2 diabetes diagnosis. Likewise, there are many such reports that debate the usefulness of diabetes in predicting diabetic complication. On the other hand, glycated albumin is emerging as a potential alternative marker for diabetes. And again, in the literature, various reports that promote glycated albumin as a potential marker for the diagnosis of prediabetes, as well as it's a useful marker in the gestational diabetes. And this particular reference says that it is closely correlated with the coronary artery diseases than 1,5-anhydroglucitol or glycated hemoglobin, that's HbA1c. Therefore, we try to understand the chemistry of albumin and hemoglobin glycation. Albumin is the most abundant plasma protein in circulation and readily accessible for glycation in circulation. Whereas hemoglobin is also more abundant than albumin, but it's an intracellular protein and requires glucose transporter GLUT1 for its glycation. Albumin is a large protein with molecular mass of 66 kilodaltons, and it has large number of lysine residues. There is 59 lysine residues and 26 arginine residues. Whereas hemoglobin is a 16 kilodalton, but it's a tetramer, and it has 17 lysine residues, but the N-terminal valine glycation is considered for the measurement of hemoglobin and for the measurement of HbA1c. The turnover of albumin is about 18 to 21 days. Therefore, it can provide the intermittent glycemic status, whereas the turnover of hemoglobin is about 90 to 100 days. So it provides the glycemic status of preceding 3 months. Glycated albumin not only provides the glycemic status, but it interacts with the RAGE and accelerates the progression of diabetic complication, while HbA1c doesn't interact with the RAGE. Currently, glycated albumin is measured at the intact protein level by enzymatic method. However, in the beginning of the intro slide of glycation, I have mentioned that glycation leads to formation of various heterogeneously modified proteins such as carboxymethyllysine, carboxyethylysine, asparaginine, imidazole, and so on. These cannot be measured by enzymatic method. So further, there's a lot of complexity exists with regard to albumin glycation, as it has 59 lysine residues and 26 arginine residues. And even if we consider only 3 modifications, that is Amodri modified lysine, carboxymethyllysine, carboxyethylysine, and if we consider only 3 lysine residues, we get several combinations. One combination could be all these 3 lysine residues modified with Amodri modification. The second combination could be all these lysine could be modified with carboxymethyllysine, and third could be carboxyethylysine, and fourth could be combination of Amodri, carboxymethyllysine, carboxyethylysine. Fifth could be Amodri, carboxymethyl, carboxymethyl again. So what I'm trying to emphasize is here, because of several number of lysine and arginine residues, as well as because of several heterogeneous modifications, it's possible that various proteoforms of albumin, of glycated albumin, might generate, and we do not know what is the clinical and biological significance of so many proteoforms of glycation. So I did a simple calculation considering at least 30 lysine residues or original residues if they are modified, the permutation and combination are 24,360 and 4,060 respectively. So, so it's quite complex. Therefore, we try to develop method using mass spectrometry. So what we did is we modified the albumin either with the glucose or glyoxylic acid or methyl glycol. Modification of glucose led to formation of Amodri modified lysine, and glyoxylic acid caused carboxymethyl lysine, and methyl glycol caused carboxyethyl lysine. These differently modified albumin were tryptically digested, and the digested peptides were analyzed using high resolution accurate mass spec, that is quadruple orbit track. And then the identified peptides by proteome discoverer were manually characterized for the glycation modification. So one of the criteria that we used was accurate mass shift in the precursor ion corresponding to modification. So here there are two mass spectrums. The first mass spectrum is of unmodified peptide, that is KQTL. And the second mass spectrum is of modified KQTL. Here you see, I would like to draw your attention that the mass of unmodified peptide is 1128.69, whereas the mass of modified peptide is 1186.70. This particular peptide shows an increase in mass by 58 daltons, corresponding to modification caused by carboxymethyl. Then next thing is we studied the fragmentation pattern of unmodified and modified peptide. So we all know that when a peptide is fragmented by CID, that's collision in this dissociation, it leads to formation of B and Y ions. From amino terminus, they are called B ions, and from carboxy terminus, they are called Y ions. If we take this peptide, KQTL, which is unmodified, so the B ions are from amino terminus, that is K, KQT, KQTA, and so on. From the carboxy terminus, K, BK, LBK, TLBK, and so on. Now, if the N-terminal lysine of this peptide is modified, then the B1 will be modified lysine, B2 will be modified lysine plus other amino acids, and all B ions will retain the B1 ion. So therefore, all the B ions show an increase in mass corresponding to the modification, whereas Y ions do not retain the modified lysine. So therefore, the Y ions between unmodified and modified remain same. So with this background, we annotated the modified spectrum. So we characterized the glycation modification of this the KQTL peptide and KQTL peptide modified with carboxymethyl lysine. So we compared both B and Y ions. Here I would like to draw your attention. As I said in the previous slide, there will be common Y ions between control and modified peptide. This is what we see here. The Y ions are same between unmodified and modified, whereas like 72 diamonds. So
Sorry for the interruption.
Good morning, all. At the outset, I would like to thank Professor Sanjeeva for giving
I'm sorry, I'm facing a bit of connectivity issues. Please bear.
this opportunity. Identified peptides by proteome discoverer were manually characterized, analyzed using high resolution identification of glucose. Therefore, we tried to develop method using mark. So what we did is we modified the albumin either with the glucose or glyoxylic acid or methyl glycol. Modification of glucose led to formation of Amodri modified lysine, and glyoxylic acid caused carboxymethyl lysine, and methyl glycol caused carboxyethyl lysine. These differently modified albumin were tryptically digested, and the digested peptides were analyzed using high resolution accurate mass spec, that is quadruple orbit track. Tryptically digested, and the digested peptides were analyzed using high resolution accurate mass spec, that is quadruple orbit track. And then the identified peptides by proteome discoverer were manually characterized for the glycation modification. So one of the criteria that we used was accurate mass shift in the precursor ion corresponding to modification. So here there are two mass spectrums. The first mass spectrum is of unmodified peptide, that is KQTL. And the second mass spectrum is of modified KQTL. Here you see, I would like to draw your attention that the mass of unmodified peptide is 1128.69, whereas the mass of modified peptide is 1186.70. This particular peptide shows an increase in mass by 58 daltons, corresponding to modification caused by carboxymethyl lysine. Then next thing is we studied the fragmentation pattern of unmodified and modified peptide. So we all know that when a peptide is fragmented by CID, that's collision induced dissociation, it leads to formation of B and Y ions. From amino terminus, they are called B ions, and from carboxy terminus, they are called Y ions. If we take this peptide, KQTL, which is unmodified, so the P ions are from amino terminus, that is K, KQT, KQTA, and so on. From the carboxy terminus, K, BK, LBK, TLBK, and so on. Now, if the N-terminal lysine of this peptide is modified, then the B1 will be modified lysine, B2 will be modified lysine plus other amino acids, and all B ions will retain the B1 ion. So therefore, all the B ions show an increase in mass corresponding to the modification, whereas Y ions do not retain the modified lysine. So therefore, the Y ions between unmodified and modified remain same. So with this background, we annotated the modified spectrum. So we characterized the glycation modification of this the KQTL peptide and KQTL peptide modified with carboxymethyl lysine. So we compared both B and Y ions. Here I would like to draw your attention. As I said in the previous slide, the there will be common Y ions between control and modified peptide. This is what we see here. The Y ions are same between unmodified and modified, whereas the P ions show an increase in modification, increase in mass corresponding to the modification. For example, here I would like to draw your attention here. In the case of unmodified peptide, the mass of lysine is 129, or that is 128 plus 1, whereas same lysine in modified peptide shows mass of 201 and shows an increase in mass corresponding to carboxyl ethyl lysine, that is 72 Daltons. So since B1 is retained in all the B ions, so therefore all B ions show an increase in mass corresponding to Cl modification. And the third criteria that we consider is MS-MS spectra should have consecutive B or Y ions. So in this particular mass spectrum, MS-MS mass spectrum, you see that we have B1, B2, B3. Likewise, we characterized 50 glycated peptides corresponding to 23 lysine residues of albumin, and then constructed a mass spectral library consisting of precursor masses of all the modified peptides and their corresponding fragment masses. So this information is very useful, especially if you want to do a targeted quantification by approaches such as MRM or PR. So next, we quantified these glycated peptides in prediabetes by an approach called parallel reaction monitoring. Parallel reaction monitoring is done on a high resolution mass spectrometer, that is Q-Exactive. It is something similar to MRM, except for that the third analyzer in case of triple quad is quadruple, whereas here the quadruple is replaced with orbit trap. So therefore, we need not have to select the fragment ions. So all the fragment ions can be used for the quantification. So the study design was, so we we did a pilot experiment wherein we took 20 healthy plasma and 20 pre-diabetic plasma, and this study was conducted in collaboration with Dr. Unnikrishnan at Cheraram Diabetes Institute, and we quantified all the glycated peptides, and the area under curve was generated using a software called Skyline. So again here, we considered three different criteria. The first criteria was modified peptide should co-elute with the unmodified peptide. So the peak area that you are seeing here, that's the peak area of unmodified peptide, whereas the shaded peak area corresponds to the peak area of modified peptides. So the same thing we observed in all the three peptides, KVPQ, KQTL, and FKT. The second criteria was the selected peptide should be present in all the replicates with approximate delta retention time within plus or minus 2 minutes. So in our study, we have acquired we did the acquisition in 120 replications. So you see here, all these, the retention time in all the acquisitions was within the limit of plus or minus 2 minutes. And third criteria was, in the Skyline software, we should have at least 3 co-eluting fragments. So this is the area under curve generated by Skyline software, and these are different peptides. You see that there are more than 3 fragments in each of these peptides, which are there are more than 3 fragments that are co-eluting in each of these peptides. So using this information, we quantified glycated peptides that are abundant in pre-diabetic condition. We found that 4 peptides, namely KVPQ-CML, KVPQ-Amodri, KQTL-Amodri, FKD-CML. So these 4 peptides showed higher abundance in pre-diabetes. And we also quantified an unmodified peptide to check whether our quantification is correct or not. So this particular peptide does not undergo glycation, and we see that the abundance of this peptide is more or less similar between healthy and pre-diabetes. And amongst glycated peptides, what we found was KVPQCML was the most abundant in pre-diabetic condition. The next thing what we did was we did the fold change analysis and compared with the known markers such as HbA1c and fasting blood glucose. So all these glycated peptides show more than 1.8 fold change, whereas the known markers such as HbA1c or blood glucose shows 1.16, 1.18, or 1.15, which means these particular glycated peptides determine pre-diabetes better than these established markers like HbA1c or blood glucose. And we also further we did modeling and found that these peptides corresponds to lysine 36, lysine 438, and lysine 549, which are present on the surface of the albumin structure and readily accessible for glycation. And therefore, we term them as glucose-sensitive lysine residues. So, therefore, these markers, the panel of these glycated peptides, could be useful in predicting pre-diabetes. So, further, what we did is we correlated the abundance of these glycated peptides with various clinical parameters. We find that these peptides correlate with the HbA1c and fasting blood glucose, as well as this shows a positive correlation with the serum triglyceride and negative correlation with the high density cholesterol, suggesting that this could also be useful in predicting a cardiovascular risk. Further, not only glycated albumin not only provides the glycemic status, but it also interacts with the receptor for advanced glycation end product. So this interaction leads to activation of oxidative stress and activation of pro-inflammatory pathways. The RAGE receptor is present in various cell types: retinal cells, neuronal cells, endothelial cells, and many other cells. In fact, we also have studied the age-RAGE signaling involving glycated albumin and RAGE signaling in neuronal cells and performed the proteomic analysis. Our proteomic analysis suggested that there are various proteins that are get differentially expressed, but I would like to here emphasize on 2 proteins, that is cathepsin B and asparagine endopeptidase. Cathepsin B has a beta secretive activity. As a result, it produces A beta levels. It increases the A beta levels in neuronal cells, whereas asparagine endopeptidase has an inhibitory activity towards protein phosphatase C. Okay, so protein phosphatase C is involved in dephosphorylation of tau. So if asparagine endopeptidase increases, then it inhibits protein phosphatase 2C, and as a result, there is a hyperphosphorylation of tau. So accumulation of A beta and phosphorylation of tau. These are the two hallmarks of AD. So in this particular study, we have shown a mechanistic link between glycation, between diabetes, glycation, and progression of diabetes to Alzheimer's disease. Similarly, in the HUVEC cells, glycated albumin interacts with the RAGE and induces endothelial dysfunction. Here also we performed proteomic analysis and found that ACE-RAGE interaction activates nuclear translocation of NFKB and regulates the expression of various proteins such as ICAM, BCAM, ENOS, VEGFR, which are involved in endothelial dysfunction. So what I would like to emphasize here is glycated albumin not only provides the glycemic status, but it also interacts with the receptor RAGE, causing oxidative stress and activation of pro-inflammatory pathways. From our two proteomic studies, what it indicates is it is involved in the progression of diabetes and diabetic complications. So with this, I conclude talk. This is a summary of. So in our study, we developed mass spectral library for characterization of glycated peptides of albumin, systematically characterized glycation-sensitive peptides of albumin, 4 peptides including FKD, KVPQ, and KQTL corresponding to 3 glucose-sensitive lysine residues, K36, K438, and K549, respectively showed significantly higher abundance in free radicals. These peptides also showed a significant correlation with various clinical parameters, including fasting blood glucose, postprandial glucose, HbA1c, and altered lipid profile. Together, these 4 peptides constitute a panel of markers that can be useful for prediction of prediabetes. Glycated albumin not only provides glycemic status, but it interacts with the RAGE, leading to oxidative stress, inflammation, and contributes to the progression of diabetes. I thank all my collaborators, funding agencies, and my students. Thank you very much.
So that was Dr. Mahesh Kulkarni for us. Thanks to him. If you have any queries, since he was not available online today, you can post it in the chat box with your name and your email ID so that we can forward it to him officially. Next on our speakers panel is Dr. Suman Thakur. I would like to ask her if she's present here online. Good morning, Dr. Suman. So I'll share my screen and start with your intro video. Thank you.
Ladies and gentlemen, we are honored to have with us our guest speaker for the day, Dr. Suman Thakur, who joins us from Center for Cellular and Molecular Biology, Hyderabad. Dr. Suman Thakur is a principal scientist at CCMB, Hyderabad. His research interests involve application of mass spectrometry in health and diseases, especially in finding biomarkers and for development of drugs for cancer and endocrine disorders. His lab uses mass spectrometry based high throughput quantitative proteomics, natural product chemistry, cell biology, animal models, and modern biology approaches to find targets, develop new drugs, elucidate its molecular and mechanistic details against cancer, investigating the efficacy of various purified and characterized natural and synthetic metabolites with minimum side effects on different cancers, including melanoma, retinoblastoma, leukemia, using in vitro and in vivo methods, is one of his major areas. Currently, he is the editorial board member of Scientific Reports and BBA Protein and Proteomics. Please join me in welcoming Dr. Suman Thakur.
Hello.
Yes, sir. Welcome, sir. So you can start with your presentation.
Yeah. You can hear me?
Yes, sir. Yes.
Can I share the screen?
Yes, sir.
Can you see the slides?
Yes, sir. We can see the slides. Kindly make it full screen and you can start.
Okay. So now I can start?
Yes, sir. Thank you.
Thank you very much. First of all, I would like to thank the organizer for inviting me. And what I'm going to talk to you about today, that target to therapeutics of SARS-CoV-2 and its variants. Now, you know very well what I will talk in this vaccine, proteomics, target, and therapeutics. First of all, this is a really very sad moment. We lost our colleague, Professor Rinty Banerjee. She was well known in the field. She was very nice person who always thinks about the science. Definitely no need to tell because this symposium is completely about her. And what was the interesting things about the things? This COVID has made a lot of people to jump on the COVID research. And she was one of that who took it very, very aggressively. But unfortunately, we lost. With this, anyhow, we all know that how things goes, how things will happen in SARS-CoV-2. We all are suffering with from last 2 years, and we don't know how long this will prolong. Earlier, we thought maybe this pandemic will become endemic and this will be end, but it doesn't look like soon it is going to be over. So this is just like a stimulation. You can see how the things goes here by and how this ACE2 helps to come inside the body. But what we do it, I had chance to be the guest editor in 2020 when COVID was in first year. At that time, we have made the completely a special issue, especially on the proteomics and its application in pandemic disease and targeting mainly the COVID. And we had that time 42 manuscripts. After that, what I'm going to talk you, because major things about the prevention of this COVID for future is that vaccine. And vaccine is very interesting because all over the people have jumped it and they have approached different types of vaccine that we call diverse vaccine platform. That's inactivated viral vector vaccine and all these things are there. So now what is the question that why different type of diverse vaccine is protecting us in different way? So can we merge it? Can we take it homologous, heterologous? Which way we should go? That I will discuss in details in later. After that, there are different proteomics has played wonderful role that especially to find the target because a diagnostic is so much costly machine till you go diagnose by mass spectrometry. It doesn't go practical. You have to take quick and so many other antigen, other tests, PCR tests are there. But where is the problem that to find the targets? Lot of people has done phospho and there are about phospho sites and phosphorus ready in SARS-CoV-2 and especially what happens to the host. More than 15,000, 20,000 phosphosites have been detected. So how to utilize all these things, that kinases as a target? That's the interesting area that I'm going to talk to you. But this SARS-CoV-2 has made so much things, so many signaling pathway has got impacted by that. And so we have to figure out which pathway is first, which pathway is second, or all the pathway are going simultaneously. That's the great question is there. That little bit I will try to talk. Then comes the therapeutics. So if you know pathway, if you know the targets, you will try to go to inhibit or activate that and try to find mostly inhibition. And that one of the area is the ACE2 you can go. And another that emerging area is coming. Now virus is not such a nice guy we can tell, because this play and change so many things. So idea has generated, why not target one inside the virus and another inside the host human test targets. Now all these approach. So basically what we are doing, targeting both virus and host enzyme together to get cure and save the life. That's the way things are moving. But before that I go on the work, I would like to talk about a little bit COVID, and whole my talk will be focused only on COVID. Okay, so in this we know very well this is the most important protein in this is the spike glycoprotein, and that's the play important role. A spike protein, membrane, nucleoprotein, genomic RNA, all are the part of such a small organism, but has created havoc in the world. Now, if you see how we'll go, this is the scanning electron microscope. You see how this nicely sit, and this replicates so fast that you have to definitely inhibit their replications. And that's the reason you can see in a scanning electron microscope, you can see transmission electron TEM, you can see that. Now, if you look that India has been impacted in different way compared to world. India has been impacted very hard by Delta. That's the time when it was the last year, April was the peak time. And then recently we have impacted by Omicron. But if you look the death number in India, majority of death happens during the Delta variant. So now the question is what was in the Delta, what we have been prepared or unprepared, and what happened, why this was so much aggressive for especially Indian population, because same variants is different. If you look about the global, you can see global situation is completely different. In global, Omicron is very high and death level, this is the area where beta, gamma, alpha also came. So all over the world, death was very high here also, but in our case, Delta has damaged more us. Similarly, Omicron has gone very high in other countries, but we have not been impacted in the case of the death. Transmission was there. So different zone, different things is impacting different way. Now you can say how hard Europe and America has been impacted by the Omicron, while South Asia, where is not impacted that much. So this looks like somehow that's management. It is very hard to pinpoint what is the things, because neither it is vaccine, neither it is things, how it separates. Why it is surprising, so many variants like Omicron has come from Africa, but this has not impacted that much in the number. So this is a very interesting thing is coming. Why this is spreading? Is it just like a regional based, or this is the zonal based? So that will be the future nice area to look at that. If you look the same way the death, this is the Delta all over the world. This is alpha, beta, gamma, Delta, and Omicron all are there. But this is our in South Asian that we can see this has been impacted majority by Delta, and especially India has been impacted here. Now, if you think about that, this is due to vaccine reason, this is due to income, lower income group, due to availability of medical facility. It doesn't able to explain any of these things. How? You can see simply how this is Africa has minimum, where very low income, low developed area is there, while Europe and America is very highly developed, but highly impacted. So this is nothing relation with the developing income and that. That you can see completely by how WHO region has there. Now we think so many vaccine has come. Vaccine is almost there from more than a year. But still you see case of Omicron is growing. This is the last 7 days of last week data, where you can see this is a still number of growing. Even your total vaccine dose administered for 100 population is about 144 is global level. Well, in the case of Europe and America, it is very high, 166, 172. But still, number are very high in last 7 days. That means there are vaccine is waning. So many things are going on. So people need to look from the different and diverse angle. Now, if you look to Africa, where number is still lost, where Omicron has developed, but number of the vaccination is also very less. So this is not directly related with the vaccinated, unvaccinated, and all these things are going on. It is very hard to understand this complex in a straightforward way. If you look the income group, this is the high income group. You see in last 7 days is still a total number also you see it is very high. Even where vaccination number is a very high. Where lower income group number of is also very less and even vaccination is less. So vaccine is not directly looking proportional. Even same income is not looking proportional. So that means what looking? Basically, it is your immunity or it is your things, how you have taken care. It is really hard to tell in one sentence. Now, if you look havoc of COVID, how it is going from last 7 days, if you look how this is impacted by in the case of US, why somehow we are better, India is better doing now in this situation, but we don't know what will happen tomorrow. But somehow where the case of COVID has originated in the China, you can see now they are picking that number and even vaccination rate is the very high. So this whole situation is becoming so complex and complex. People thought it will be slowly becomes endemic, but it doesn't look like. It looks like it will be prolonged sometimes, and we have to be really careful with the mask, and we have to really careful with all the behaviors. Now with this, just I will talk you before going to that, why this is able to do so nice. So you can see most dangerous variants was the Delta. That doesn't have that much mutation, but concerned mutation was the K417N. That was the clear cut. While all these mutations are also present in the Omicron, but it is not going to that much severe because a lot of people are vaccinated and a lot of people are things. But still, this is transmission level is very high. Now you can see this is the variants of concern today. It is the Delta, it is the Omicron, and BA2 is the major. Transmission level is very high, immunity is very better damaging, but severity is low due to several factors. So these are all the data is coming. And now recently what XE variants is coming, that has got also mutation of L452R, F486 and 346K. So this Omicron is making so many recombinant and so many variants are making combination, and that's becoming a really big challenge for that. What is the danger? It may be not mutate and become so much problematic in future. With this, just you see, now you go to the structural level. If this is the domain of SARS-CoV-2 spike protein, you see this RBD level where it is the wild type. Delta has 2 mutation and now it has become so much. So it is really that SARS-CoV-2 is changing themselves very fast mutating. Their major target is the immunosuppressed person or someone even no one is knowing, and it is mutating inside the human body very drastically. With this, you can see the 3-dimensional structure. Its Omicron is the full of the mutation in a spike trimer, while there is a very less even in the most dangerous Delta. So now mutation is becoming the major point where they are changing the things in which way. This is that you can see Delta RBD and Omicron RBD. So this is a structure level, lot of changes are here, hoping that this will be slowly kill the virus. But it doesn't look like that what we thought earlier. Now, if you go by symptom, it is hard to tell by one symptom this is there. So you have to be, earlier we had loss of a smell, but this is not less common in the case of Omicron. Now, this is the phylogenetic tree is coming. Now, how the things has been gone. Now, Delta, where was the most dangerous, that was the hair at this place, but Omicron has developed parallel. So what happened in this whole world scenario, different variants are coming, different place, and they are growing and making a different area. Even Delta and Omicron is not at all connected. That's look. Now, what I will talk to you about the vaccine, there are about 8-9 vaccines has really fully is used in the world, and now they are protecting a lot of people in that. AstraZeneca, Johnson Johnson, Moderna, Pfizer, Sinopharm, Sinovac, Covaxin, this is our Indian vaccine, and Covax, Nuvaxovid, and Covishield, that's made by AstraZeneca and made by Serum Institute of India. So Covaxin from Bharat Biotech and Covishield from Serum Institute. This India has done great part in this lab. But scientific question comes when you have different type of this vaccine. This is different diverse platform, how you can use, and their mechanism is different. Inactivated works in different, viral vectors works in different, especially in viral vector groups is the COVID, Covishield, Sputnik, Ad5, and Janssen. Now, if you look about the inactivated, where our Covaxin is there from Indian vaccine, that BBI, all this looks indifferent. Same way protein-based vaccine is look different. RNA vaccine, that's from Moderna and Pfizer that came, it works in different way. Now the question is, if all are working different, how you think about the using one after another? If you use another one after another, right, two diverse platform that calls heterologous. If you use same diverse platform, okay, then it is homologous. Mostly till date in India, it is homologous is going on, but accidentally it happens in the 18 case in there where they have injected first dose as a Covishield, second dose as a Covaxin, that works better. So this area is growing very well. That will solve the vaccine different way, especially it will improve the immunity. Now, basically what we are talking in future, it will be homologous or heterologous. Now, vaccine platform under emergency use, majority are inactivated virus, and now this nascent technology that has grown very well, mRNA is coming up very well. If you look the clinical trial, major clinical trials, this data is every day changing, and this phase 1 is turning towards the phase 4. That means, but in phase 3, if you are approved, this generally get from emergency use. So a lot of vaccines, lot of effort all over the world is going, and that is showing how things are moving forward. Now, if you look mode of administration, that's the area is lot of research is going on, because most of the vaccine, whatever we have taken and all the audience would have taken, it will have been intramuscular. But future is coming towards intranasal. So that's where upper respiratory part should be blocked and infection should be reduced. So that's the area what it is coming on. And now there are oral is also people are trying. Let's see how the times is going. And if you look vaccine platform in clinical trial, major trial is coming from protein subunit. So many vaccines are looking now by targeting protein subunit that way. This is very nice things. If you take different diverse platform, if when especially variant will emerge, this vaccine will be very useful. Maybe one will be not effective, other will be effective. That we can test quickly, and we should push that type of diverse vaccine platform for particular that type of variants. So this way we are showing on vaccine platform in preclinical trials. So many are there. Can you see maximum number of preclinical trial is going through the protein subunit. So that's the area is coming very nicely. Lot of vaccines are under pipeline and things are moving better. If you talk about the dose, mostly we have gone through 2 dose, that is a 0 plus 28. Now this dose also matter, and people are changing 28 to 56, sometimes 12 weeks. We all have seen in India how Covishield dose has been changed for 12 weeks. So now this dose is also matter, because where it will play the booster dose and where immunity will be improved. Now, real question this has set that homologous or heterologous vaccine. Now, homologous vaccine, I have told you, if you take same vaccine, 2 dose after particular interval, that's called homologous. If you change the diverse vaccine platform, just like you are taking viral vector, now you take second dose mRNA, and same way mRNA second viral then inactivated, or inactivated and then viral, that's called heterologous. This is the area is coming in the future that will be very nice after some time. That is protecting very, very well. Especially we have seen in the Europe side where clotting problem was there. After giving AstraZeneca first dose, they have given mRNA vaccine that works better than homologous vector and homologous mRNA. So heterologous has future, it looks like, but it will take time to establish and time to come in that, because you need details, complete clinical trial before that. So that's, I will give you one of the example, just like this homologous mRNA vaccine, they do better humoral immunity, and then homologous vector vaccine, they give better cellular immunity, but both gives all the other. If you merge heterologous, that means vector after mRNA. This combination is working very well. You have both humoral and both cellular immunity, and you can able to protect SARS-CoV-2, especially in the case of variants, better. That was the whole idea. That's the whole things is the coming in the future. I'll give you, there are so many clinical trials happen. One of the clinical trial example I will give you that came in the NAGM, where they have given primary dose for the primary dose they have given Pfizer, that's mRNA. Then they have given secondary dose with Moderna mRNA and Johnson Johnson. You can see even both are mRNA vaccine, but still, if you give just like homologous BNT Pfizer mRNA with Pfizer mRNA, you are able to increase the titer. That's the factor increase about 15 to fold. If you give same platform, different vaccine, that also able to increase better in this case. So it is showing when even diverse vaccine platform, if you are using two type of vaccine in same platform, also that is helping very well. Now, but if you do give this combination with mRNA after that vector, that's the Johnson, it doesn't work that much way. So all heterologous will not work. We have to see permutation and combination, which one is working better. Similarly, if you give primary mRNA Moderna first, then you give Pfizer, this is working far better compared to homologous of Moderna. So this is again interesting that even in the same platform of vaccine, two different company vaccine is working better. While most interesting is that if you give vector first, that means vector-based vaccine, anything you give first, and then you give second mRNA, this is increasing, factor increase tremendously. It's very wonderful. So what results are coming? Vector-based vaccine first you should give, and booster dose you should given by mRNA. Similar result is coming. Vector-based vaccine should be given as a primary, and inactivated vaccine can be given secondary. Example is Covishield first, Covaxin is second. Same example is there. So many places AstraZeneca first, and then mRNA vaccine second. So what new completely theory is coming that heterologous vaccine is working very well. Not all permutation and combination, particularly things are working, and especially where mRNA is the booster dose. Now with this, I will talk about little bit proteomics, and you all are very familiar. All of the world people, whoever has facility, whoever has things, they all have jumped on proteomics. They try to contribute whatever, small or medium or big, whatever you can contribute. Everyone has attempted. That should be appreciated. That's gave the lot of research going on in the proteomics. Now, you all are very well that area we can do post-translation modification, interaction, quantitative proteomics, biomarker, therapeutics, all you can do, and all has been utilized. People can go to deep proteome. Even we are going to show you 15,000 phosphosites has been detected by people, and even you can detect 1 or 2 protein also, a spike protein, that also made research. So what way you are doing research, that's mattering. Either you can do protein chemistry or you go big number of proteomics also you can go, but all are contributing to COVID. This is definitely, I think most of the people are knowing mass spec very well, but if any new students are there, few slides I will tell you basic, because without basic, after that it becomes Excel sheet and big data and number, you can't test it. Basically, mass spec is just weighing the molecule. It's very simple that you can figure out. And when we inject the mass spec, inject the molecule, inject your sample, it goes in positive ion mostly, and we see multiply charged positive ion, and we take that fragmentation. That's the reason where you do the things. Now this, after that, basically what you do, nature has made in this way. First they made amino acid, then they made peptide, then they made polypeptide, and then they finally folded the protein. We do completely opposite way. We take the protein sample, we take the completely crude extract, we completely use the proteolytic enzyme, we unfold it, we cut it, we chopped it, and finally make to peptide, and then fragment and do amino acid, and then we sequence. That whole problem is coming the bottom up approach. That's called, we call shotgun proteomics. That's way. Basically, what this is the molecule where you are giving the energy and you are breaking in this the piece, and you are combining both the piece and you are sequencing that. So this is the very basic things I just told. After that, this is becomes like just table and number game. There is no basic things you need to know. proteomics tools to understand biology. But if your basic is clear, then you can understand what is happening inside the mass spec and what is your result. Aim, sample preparation, HPLC chromatography, mass spec detection, fragmentation. Mostly we are talking about here, but most important is you should have aim, you prepare sample very well, you do chromatography very well, and then you analyze very well, because after that everyone is going to see your analysis result. This is the things, you take tissue, cell, lyse, proteins, peptides, make proteins to peptides. I told you, use any tryptic enzyme, lyse, glucose, any proteolytic enzyme. You can do fractionation more or less, whatever according to your health and wealth. And your setup of the mass spec, reverse phase purification, nanocall, you can use mass spec. There are so many collision energy site is there, CID is there, HCD is there, ETT is there. And then you can do label free and that. And finally, you identify your proteins using the software. That's the whole schematic I gave you. And after that, similar way it is the metabolomics, but we are not talking today metabolomics, but similar we can do. Now I come to what's the result is there. There are so many labs in the world has tried to figure out that complete coverage of the COVID that. Now, if you look about that, more than 15,000 host phosphosites has been covered. What found the interesting, SARS-CoV when enters the body, they triggers the phosphorylation very big. So viral protein phosphorylation, we have attempted, we have taken all the data from all the published and we have analyzed, we have done, and we found about 71 virus phosphosites we have reported. And this kinases we have seen about more than 15,000 host phosphosites people has reported. We have taken all the data, we have combined and we have analyzed. What came in interestingly, this kinases is playing big role, and this kinases can be as a target and therapeutics. How this is impacting so many signaling, MAPK, GFR, TGF-beta. Can we block this signaling and think about the therapeutics? That was the whole aim. If you look about that, about the 71 phosphosites, nucleoprotein that's inside the SARS-CoV-2, nucleoprotein as the major phosphosite has come. And that also came in the majority in serine and less in threonine and very less. But when a spike protein, we have got phosphosites reported in the tyrosine. And when we analyzed the data of several people, that we saw that it is coming regularly. Now, if you look about these all phosphosites, what is the most important? That's NSP3, NSP6, and in a spike protein, there are so many phosphosites are there. If we do more deeper, maybe we get more deeper. That maybe because you are doing a one time point, other time point, we are missing that. So different time point, different things needs in future to do that. But at 71 phosphosites in SARS-CoV-2, that is itself a nice data we have analyzed. If you look all the phosphosites, all those things, these all are coming with different mechanisms. Here is the things where you can target. Impact of SARS-CoV-2 is such high that if you enter through ACE to take help of the TMPRS2, but there are so many pathways there, especially this PIK5 can be taken, this RAS, RAF, MAPK, MAP2K pathway is there. This is PI3K, AKT, mTOR, CO. All this has been completely phosphorylated, has been caught in the host. So now one side is the completely theory is going inside the virus, another inside this is the havoc of change which is coming, especially P38 is there, and another pathway is the coming JAK. So now this new target have inhibit the JAK state by using baricitinib. That I will talk you later. And then you see how this JAK you can inhibit, and this one by one you can inhibit any phosphosites. What is the green? This all has been detected very nicely. Now several signaling, several pathway it is going because whole things are changing. Cytokine storm is coming. Now these things looks like can be explained, but still it is not easy. Now if you take all this, that heavily phosphorylated protein, and then you see the interaction, what you find, it's all are interacting very nicely. That means whole chain are interacting, whole chain are there. If you inhibit one, there will be trigger of other place also. Same things happen. These are the, you can see about 15 compound that is going to target to find the therapeutics for the SARS-CoV-2. And this 15 is targeting in the host, that which place. I told you about PIK5, AKT, MMP2, TGF-beta, all these things are targeted. These are the already under clinical trial for the COVID drug. So all these targets are very nicely coming at one place. Even EGFR people are targeting. Basically, these all are the repurposed drug. We know this compound. We know this compound is targeting which that. And when we see in that COVID, this is coming and this is getting phosphorylated, this is getting upregulated, this is heavily phosphorylated, then you target this and take. Different company, different lab, different country has taken these targets and doing clinical trial. But if we have taken all the things and we have analyzed and we saw nicely this is interacting. So that means one place if you are impacting, other will have also impact. So this way therapeutics is going on, and that shows the future how things will be there. Now, if you look SARS-CoV-2 protein, I told you in nucleoprotein how this is the completely phosphorylated. Especially this is the chain where highly phosphorylation of serine has been detected. So that's matter. Nucleoprotein is there phosphorylated, and if you look that number of serine phosphosides are higher compared to threonine. So that's the logically we see in other sample also in cancer other. Most of the time serine is getting heavy phosphorylated. Same things if you look about the spike protein, that is the most important. You see here tyrosine has been gone phosphorylated, and that we can see here clearly. So serine is phosphorylated, threonine is phosphorylated, and tyrosine is also phosphorylated. But majority of combination is the serine has been phosphorylated. Similar data, you look at SARS-CoV-2 protein in membrane protein, you can see where it is completely 4,4 serine has been phosphorylated in the case of membrane protein. ORF9 protein we can see ORF3, and all this has been caught by different studies. So this shows how the target, how the things we can find. Now we talk about little bit therapeutics, a status of COVID-19 medicine. Majority of the medicine, that's the dexamethasone, remdesivir, this molnupiravir, and is in the use. But still we don't have sure short medicine that in every stage, just like pre-stage, hospitalized stage, severe stage, and very severe critical stage, we can able to treat. It is better to prevent this disease as early as possible. These are the has been approved by World Health Organization, and these are the already in the use, but still need to find lot of other things. Now, what we are trying to find, the ACAT analogs can be used as because there are a lot of data and things are there. So captopril and these all can be used. So a lot of study is going on this way. Let's see how the future is turning. But what is the most important things I am going to talk to you now? Why target now at simultaneous host and virus, both together? That is the future, because this virus is not easy to control because he is damaging host also. So in target virus, this is just like remdesivir. Remdesivir is the prodrug. When you put inside, it is changing GS5, then alanine metabolite, then nucleoside, and this is nucleoside triphosphate. This mimic the ATP and this bind to the RP. So RNA-dependent RNA polymerase get inhibited, and finally viral replication is getting over. So this is the mechanism of remdesivir, mechanism of antiviral things are there. Now we have to see how we can see that. So now this is the virus enzyme. I explain you that's the remdesivir. This is the alanine metabolite, changes in the structure, now nucleoside monophosphate, triphosphate, and then this is the SARS-CoV-2. This is happening inside virus. Now you see how you can do. You can take the varicitnate. You know very well that JAK has been highly phosphorylated. JAK has changed. You can inhibit the JAK, JAK enzyme. So at a time you are inhibiting in the host enzyme, host also. At a time you are giving in the virus also. So combination of remdesivir plus baricitinib is one of the example that combinatorial drug can be very useful. So what you are doing in COVID-19 treatment, targeting both virus and host enzyme simultaneously. That's the very important thing. That is the new strategy, and it looks like be successful, and lot of clinical trial is going on, especially when today's days remdesivir with baricitinib with maripavir, DWJ, lopinavir, and this has already hydroxychloroquine is out from the market for COVID, but a lot of people has started with see that it is there. Definitely this will not be useful, but clinical trial has been started. So just I wrote down lerizumab, and their reason is their tocilizumab, interferon beta 1a, interferon beta 1p. So all these compounds are there. they are doing combination with remdesivir with other. So that means target both virus and host enzyme. All are under clinical trial. This is the another way going to be hope. Now, if you look about the baricitinib, this comes inside the body and they inhibit JAK1, 2, GAK, AAK. This cause endocytis. This go for cytokine signaling and inflammation. This finally these two go also help in the viral stoppage. So this is the targeting host enzyme, and then another is the targeting virus enzyme. These two together, if you look how things is going in that body, so remdesivir virus-based target and baricitinib human-based target. Now this combination can be changed. Remdesivir can be used with other human-based target. That will give you a great sign of the life. Now, with this, if anyone has a question, you can talk. We had also chance. Just I would like to show you a couple of slides where we can use how the proteomics has been used that all over the world. In this issue, we have the chance to publish that. Especially this was the things where just you take gargle and you put in the sample, take the sample, digest it, and put in the mass spec, and you can see that simply such a nice spectra of patent is coming. And this has come from the Germany Ealing group. So this had published in that issue. So what I'm trying to tell, this virus is flying inside the mass spec very well. Even people has developed different techniques. Just like this is nasopharynx, you can take infected patient and you can get all this nucleoprotein identified by just doing the mass spec LCMS. Even people has developed in 3-minute mass spectrometry window. That's you can take a nasal swab and peptidome you can put and you can get some idea how that is. So this all people has tried their effort and all these things has gone very well. Now you can see all the things is there. You can get completely peptide in HPLC, which place it is COVID peptide is coming. That's all help you to diagnose, confirm, and study for further if you want to grow deeper study. Even people has taken serum proteomics, they have done coagulation and complement status. That's way things has gone. So this is a just example I am giving you how the people has done, what we have done. I have shown you and how we have analyzed. This is the another thing. People have used NMR and mass spectrometry also. There they have got also the things. So this is the diverse picture of using. You can see such a nice. They have got the plot. They have done all the things. Even people have used proteomics and metabolomics together to find out the COVID-19 patient sera, get some marker, early marker, or some target information. So this all things has been done, and it's interestingly people has tried to find the interaction also in that way. So dysregulated proteins, you can see that especially when you talk about the COVID, CRP is the very important, and always this is available there. Now, same way they have done a metabolite study also, and then you can see completely dysregulated metabolites, especially you can see completely serotonin, sphingosine, all are coming in that color. People have tried also multi-omics data, resolves a sharp religious state shift between mild and moderate. So how can you differentiate mild and moderate before this is there? So try to find the early marker. This work has been done by others. Okay, so this all I try to find. A lot of people has done plasma proteomics and metabolomics. That's also even single cell multi-omics has been done for the COVID. It's really going very nice. You can see this graph and this. Its map visualization completely people has done in different way. Analysis of 50 plasma metabolites they have regulated. Proteomics of circular infected host cells reveals therapeutics. So what I showed you the example how to go, but resources, all these things has been done by people lot in study, and even they should do complete 11x MS sample that you can see. So just I am trying to show you how proteomics field has contributed, especially to the COVID, that it is there. Now, if you look about this, what they have contributed, these are the target they are telling you it has contributed. This target can be taken and they can be used in future to develop it. It is already in use so many. Even you can do protein interaction map, reveals drug completely repurposing. That's has been done by Gordon et al. And then you can see there are 26 SARS-CoV-2. This is one of the first paper that has come and that shows how the things can be done for the COVID very well. Now, this whole has been gone very well. With this, I would like to tell Thanks to all the audience. And if you have any question, I'm very happy to take you. Thank you.
Thank you, Suman, so much for the wonderful lecture once again. And my team will now convene the session and we'll ask some question. And
yeah, I'll be happy to take the question.
Thank you, Suman sir. So there is one question from the audience. It's from Garima. She asks whether targeting host enzymes such as JAK would have an impact on general physiological function of the enzymes in like in healthy humans also.
Yeah, of course. See, this is the place where you have to think very well. One side, JAK inhibitor, that's all this baricitinib, all have been used for rheumatoid arthritis. These all the compounds, what I have talked, that we have seen in this way, that all are repurposed drug. Already their studies have been done. Whatever is their side effect, that is also very known. And whatever is their merit, that is also known. So most of these compounds are repurposed drug, well-studied drug. Of course, they will have some side effect, but these all are under limit. Okay, so in any compound you put in the body, there is a little bit plus and minus, but all should be little bit come under the safe. We call there is no adverse events. So if no adverse events it is there, then it is a safe. So of course, baricitinib is safe. It is used earlier as for rheumatoid arthritis and so many other places. So this JAK inhibitor is there. But what is the interesting thing you have to see? By targeting only RDRP things, virus, you are not able to control the things. So if you are one side, you are controlling inside the host also, one side in that side also, then it is giving more benefit. So this is working. It is working better than remdesivir, especially in severe case that. But others combination now will come that will have more benefits.
Thank you, sir. Sir, I'm Medha, as I have introduced myself earlier. Sir, I'm from Professor Sanjeeva's lab only. I would like to ask one question regarding your talk. Sir, as you have given us a lot of insight on how vaccines are working for the COVID scenario and how immunity is more important as compared to other sociological factors that usually people consider for such communicable diseases. Sir, could you shed some light on whether although like most of a major of the population has been affected with COVID now, so we do have certain amount of immunity and we have got vaccinations or booster doses now we are getting. So what will be the effect, or what are we expecting in future corona variants? How is it going to affect us in future? Like, so anything you have?
Yeah, it's a very nice question. It is practical question. See, what is the things when someone listen about the vaccine, especially when you are in the science, you should know this vaccine is not going to work for you lifelong. So when vaccine has been injected, that's called vaccine life, and another technical term is the waning for vaccine. So if you look about generally you have when you give primary dose and then booster dose, average is most of the place 28 days. Is that? So booster dose is increasing your immunity. But even booster dose when you have given, average after 4 weeks to 16 weeks, generally we tell about 16 weeks to 4 months, your waning starts, that your immunity of this vaccine is going slowly down. That's the reason this theory comes that every vaccine waning is different. Like you have taken vector-based vaccine, just like our Covishield is vector-based vaccine, and Covaxin is inactivated vaccine. Every vaccine waning, that means their efficiency is going down after some time. Now the question is when? Question is how much? So now your efficacy, your immunity, all things due to what you got by vaccine, it is also going down. Very interesting result has come, particular to your question, that if a person has got COVID, generally we see that's in chicken pox and all these things, people get immune for lifelong, you will not get next time chicken pox. That is the general belief. This doesn't work in the case of the COVID. If a person has got COVID, they got immunity, but that immunity is also getting slowly waning after that sometime, just like your vaccine efficacy is going also down. So now what is the benefit? So now similar study, I have not presented that, but similar study has been done, clinical trial has been done, where a person has got COVID When a person has not got COVID, both has taken, both has given primary dose, both has given booster dose. So when primary dose has been given, their immunity, the person who has COVID or who got earlier infected with COVID, their immunity was better. Okay. But when booster dose has given, after 4 weeks, there is no difference between person who has got COVID and person who has not got COVID. So what is the meaning of that? So that means 4 weeks you take this and 4 weeks you take this. So about 2 to 3 months, there is no difference a person has got COVID or not COVID. So general belief what we had in other viral, chickenpox or other, once someone get disease, next time they will not get. This is not working in this side. So there is clinical trial proof has come already that after this only maximum effect you 4 to 6 weeks give protection. After that, slowly it is gone. So therefore, the person who has got the infection, they need also to take the vaccine. They are safe only for 4 weeks. Maximum they can go to 8 weeks. Okay. After that, waning will be down. That's the reason now new variant is coming. It is infecting a lot of people, but still some immunity, humoral or cellular immunity is there in the body that helps to little bit you, but it will not help you lifelong. That's the reason booster dose is very necessary. That's the reason whole over the world it is there. So infection is helping you first 4 weeks. After that, there is no effect on that. I hope I have answered your question. If not, you can ask second part again.
No, sir. Yes, I find that you have pretty like covered everything that I asked. But
There is a
a follow.
really what you told about social. There is a really no social impact. I
Yes.
have shown you that's low income, high income. High income is getting more infected. You have vaccinated a country that is getting more infected.
Yes, yes.
Okay. So this is nothing. Whole variants are Omicron is coming from Africa, but you see African that's per side. The number of death and case is also very less.
Yeah.
So that means
Yes.
how body is telling, how body is developing. This all are really now it's coming to whole immunity. So only everyone has to keep immunity either by vaccine or either by diet or either by exercise. No one knows this now. But everyone must have to take vaccine because exercise or diet will not save you from this.
Yes, yes, sir. So, yeah, we conclude that vaccine is a must. But, sir, as follow up to the same question is like, is there any connection that you have seen among like people having immunity and them having the severity of the COVID that they have got, like the severity of the symptoms? So, is there any connection between the two?
Yes, there is a connection. See, because one of the I have not presented, but one of the study has come. A leukemia patient was there. Okay, leukemia patient has given primary dose, no immunity. Secondary dose, boost. Secondary dose, just we give generally. Second dose, no immunity. So nothing has developed. Person is immunosuppressed. Leukemia is there, already comorbidity is there. You are giving first dose, nothing is happening. Second dose, nothing is happening. Okay, when you check their antibody, neutralizing antibody, nothing is coming. When they give a third dose, okay, that was just a clinical trial, what to do in this case. Third dose when they have given, then immunity has boosted. So what is the meaning of that? Generally for a person, first dose create little bit immunity, second dose create you majority of most of the immunity, but if person is immunosuppressed, then for that 2 doses are nothing. So they need additional. So it's completely now it's going in this way. Dosing is also important. Not only time interval is important. Dosing is important. How much amount you give, how many dose you give. So a person has comorbidity. That means they have less immunity. They need more doses to build up the immunity. Another way, now come to your question, a person has very high immunity, then they get severity. Chances is very less. Now its question is which type of immunity you have. And you remember in the case of especially Delta, what was the problem? It was a viral load. It was not only that the case viral infection. If you got highly viral load, that severity will come and a time will come, your body will give up the immunity completely. So if viral load is also important. 90% case, it is if you have good immunity, it is not going to that. But you have to understand that how much you have got infection, how much viral load you have got. If you have got too much virus inside, then you need too much immunity. Same immunity is not going to be sufficient. That's the reason some case, even with little bit good immunity, Also, people you have seen in the case of Delta, young age people also have so many died. Okay, because viral load was very high, and that's video. But to the point, this can be tell in a broader line. To the point, it is very hard to tell. How can anyone, a normal person, tell you how many virus someone has got it? Till you know full checkup and all, and neither you know how much immunity you have. Okay, so practically it is very impossible to every person should have knowledge how much immunity is there. But the general question is there, you should have good immunity and try to have minimum viral. If viral load is very high, then body has to fight very hard. And problem with immunogenicity, sometime body give up. Okay, the body can't fight too long. That's happen in all the case of sepsis. You know, sepsis shock, all this microbial agent cause, and body give up immunity. So this will happen soon. If viral load is very high, then problem is there.
Yeah, thank you, sir. Thank you for taking our questions and the very knowledgeable lecture. So if anyone else has any questions, or else we can move on to the next speaker.
Yeah. Thank you. Thank you very much.
Thank you.
Yeah.
Thank you, Suman, once again for.
Yeah, thank you. Yeah, it's very nice.
Nice
Yeah.
to see you once again.
So next we'll have Dr. Michelle Hill's talk. Due to some glitches, she is not available online, it seems. We will be now presenting her recorded talk. If you have any queries related to her talk, please put that in the chat box along with your name and email ID so that we can forward it to her officially. Thank you.
Ladies and gentlemen, it is our privilege and pleasure to have Dr. Michelle Hill with us. Dr. Michelle Hill joins us from Queensland Institute of Medical Research, Berghofer. Dr. Michelle Hill is an associate professor. She obtained her PhD at the University of Queensland on insulin signaling. She used Phosphorylation extensively during her PhD work. For her postdoctoral work in Switzerland, Ireland, and at the Institute of Molecular Bioscience, University of Queensland, Dr. Michelle Hill focused on various aspects of signaling using subcellular proteomics. With a passion for translational proteomics research, she established the Cancer Proteomics Group at the University of Queensland in 2009. She also initiated the Queensland Mass Spectrometry Symposium to encourage local networking. After successfully developing clinical proteomics research and establishing a proteomics amyloid type testing for the Princess Alexandra Hospital in Brisbane, Dr. Hill moved to her laboratory in QIMR Berghofer in 2017 to broaden her research program in precision and systems biomedicine laboratory to include lipidomics, metabolomics, and chronic and mental health conditions. Dr. Hill has been an active member of the Australasian Proteomics Society since 2007, being the Queensland representative on the board for many years. Dr. Hill has also been an elected member of the International Human Proteome Organization (HUPO) Council since 2016 and served as the Secretary General for 2019 and 2020. With these words, we welcome Dr. Michelle Hill.
Great.
Wonderful. So thank you, Sanjeeva, and thank you, very happy to be here to share with everybody the clinical diagnostic research that we have been doing, and also I will share some of the results that's quite recent as well. Just jumping into it, today I would like to share two different topics. One is the use of shotgun proteomics for clinical diagnostic, and this was initially already mentioned a little bit, the diagnosis of amyloidosis type. And the second topic, which is a little bit bigger and a little bit earlier in the development, is the use of a pipeline to discover early cancer biomarkers. So first, I will introduce the diagnostic question in the first topic. So amyloidosis is a protein aggregation disease. And this is where proteins aggregate and deposit in different tissues in the blood. So you might be aware of Alzheimer's disease, which also has amyloid plaques in the brain. So this one, the system's amyloidosis is a little bit different. So it occurs in the blood and it can deposit in many different types of organs. And when there's a buildup of these amyloids, it causes tissue damage and it could be fatal. Unfortunately, diagnosis for this disease is quite difficult because the symptoms are often non-specific. However, it is very important to identify exactly which proteins are actually depositing. And so it can occur in more than 30 different types of, with more than 30 different types of proteins that are being deposited. So what's currently done to diagnose this? Why is it very important to identify the treatment type, the amyloid type? So it is very important because the treatment is very different depending on the type of amyloid. And the treatments are very severe, so you don't want to get the wrong treatment. So I just put here two examples. On the left side is AL amyloidosis. This type is probably the most common systematic amyloidosis. It is due to the deposit of immunoglobulins and due to bone marrow plasma cell proliferation. So it is kind of a type of a cancer related. The treatment of that is to give a bone marrow transplant. On the other side, on the right side here, the ATTR is transthyretin, and there are two different types, if it's a wild type or if it's due to a variant gene, which is a hereditary type. And in this disease, then this type of amyloid, the ATTR deposits in the blood. Again, the treatments are different depending on which type it is. So for the wild type, there are some drugs that you can be used to stabilize the structure of transthyretin. There are some side effects for this, and the use is therefore dependent on how severe the patient is. For the hereditary one, the treatment was liver transplantation. So again, it's very severe treatment. New therapies are now being developed, which is RNA interference to reduce the transthyretin mRNA, and therefore there's less of the protein to be deposited. So this shows that it's very important to diagnose the patient correctly as to which type of amyloid it is. So currently, how is it diagnosed? So it's a multidisciplinary method with the clinician of different specialties taking the detailed clinical history, family history, and then the pathologists, they use the dye called Congo red to stain. It recognizes a particular amyloid fibril structure, which is shown on the picture here. It's the pink stain on the left using light microscopy, which gives the fluorescent birefringence in the right side. So that identifies in the tissue amyloid fibrils. Then to identify the actual type, antibodies. Now, these can only be done in particular known amyloids, for example, if it's a
Great.
Wonderful. So thank you, Sanjeeva, and thank you. Very happy to be here to share with everybody the clinical diagnosis of PE tissues and did laser capture.
Sorry, just
of immunohistochemistry protocols for all those and the expertise. And so this is where proteomics could come in, because we're talking about deposit of a single protein. It theoretically should be a very good application of proteomics to diagnose what is this protein. And so this method was then first reported by the Mayo Clinic in 2009 in a research paper. And the method they developed is a laser capture microdissection from Congo stained formalin-fixed paraffin-embedded tissues. And here I show a brief sketch of what it looks like, which we have these other pictures from my team. So the tissue stained with Congo red, and you can see a little example here with some red colors you're seeing. And then pathologist does a laser capture. And this then is, you can see an example of exactly how big it is. It is very, very tiny on the cap. It's the very small part in the middle, so less than 1 millimeter. This then is undergoes tryptic digest using standard methods, and then tandem mass spectrometry, so LC-MS was done, and then database searching is done. And this is bit where it is not 100% scientific yet, as far as I understand, in different centers, and at least in our center, the protein list is then reviewed by a clinician who then look to see, usually it's one of the top proteins, which is a known amyloid protein, then it can be diagnosed as that according to clinical characteristics of the case as well. So this is the current procedure, which was initially established by the Mayo Clinic. A second option for shotgun proteomics uses subcutaneous fat aspirates. And this was first reported by the University of Pavia from Italy in 2012 paper. And this is also used in different centers around the world. However, the laser capture method is the most widely used. And actually, this has already been reported from many cases, many centers around the world. And here I show a world map of the papers that were published between 2016 and 2020 that used the laser capture microdissection coupled to shotgun proteomics in amyloid typing. And you can see it's spread around the globe. Mayo has done the most number of cases. They actually have a reference lab where people can send their requests for diagnosis. So I will show a little bit of result from our study, which is done, of course, in Brisbane, and we don't have such a large number of samples due to our smaller population. But as a summary of all these studies, there's a very high identification rate, even though we are looking at very tiny amounts of tissue sample, which was already fixed. So this is a challenge initially for proteomics. But the very high identification rate was 90 to 100%, except for the Korean study, which only did a very small number of cases, and their focus was on changing the method to a targeted proteomics method. For that, they reported 100% identification. But still, in comparison, the immunohistochemistry identification rate was even lower. So the question we had in our study actually is, what is the clinical impact? So when we did this study in 2016, this was still not a yet known question. actually, that's when it was published. So the study was a collaboration between the hospital, Princess Alexandra Hospital, and the proteomics lab of the UQ Diamantina Institute, where my lab was based in, and the TRI proteomics facility. And I'll just use this as an acknowledgement for all of the collaborators involved. So in the study, we captured all of the 138 cases referred to the hospital between 2010 and 2014. And yes, it is not a very common disease. And of course, our region is a small population. That's the number of cases we had. 35 different organ sites were analysed. And so we collected the FFPE tissues and did laser capture microdissection. Some of them already, when we received it, when you're looking at clinical diagnostics, there was no tissue actually that was sent, so they could not be attempted. However, for the remaining 129, we had a 94% success rate to identify the amyloid protein. Of those, 15 of them needed a repeat analysis to identify, and 106 was successful in the first run. So our success rate in our centre was similar to the global rate reported. In comparison, the standard immunohistochemistry was attempted on 88 cases. So some of them, based on the clinical data and based on the number of immunohistochemistry protocols we had in the hospital, they were not attempted. But you can see there the success rate was only 44%, and also there was 6%, which we consider as a false identification when we compare to the proteomics results. And so this then is the significant DART statistics that came out from this study. So when we compared what would have, if there was no mass spec, what was the previous diagnosis based on existing methods, and then compared to the results from the mass spectrometry, actually the clinical diagnosis altered in almost a quarter of the cases. So put it another way, by our hematologist actually interested to implement this novel technology and to test this novel technology, he actually saved a quarter of his amyloidosis patients from receiving the wrong treatment. So this is showing a very strong impact of proteomics in the clinical diagnosis. Many centres have now implemented this technique. However, in our centre, we still have to obtain regulatory approval. So currently it's a lab developed test, and therefore it's not a recognised diagnostic. In different centres, it's a similar situation, but one of the downsides for that is actually nobody can actually be charged for the test because it's not an approved test. So that's one of the areas we need to work on to get approval and then to be allowed for reimbursement through the national Medicare system. Another area is because now many regions or many centres are setting up, different centres in different countries have different protocols. There is not really an international standard. So one of the goals is to set up an international standardization and harmonization framework. And finally, we also wanted to consider some sort of outreach activity and to enable global access to this technology. So in our centre, we already receive referrals from all throughout the Australian states. We're the only Australian centre that offer this test, but there are from other countries which they don't have such tests. They are also trying to contact us for testing, but at the moment we're not able to provide that. So from our own study, the research was really driven by my clinician collaborator, Dr. Peter Millay, and was then funded by the Hospital Foundation and the Pathology Queensland. So this is an example of the existing application of proteomics in the clinic. Now, the next topic is a little bit longer and still in development. So when we talk about research and translation, to the clinic, it is fairly well recognized that there is a valley of death when one tries to do some basic research findings, which we think, okay, usually you start already with a clinical question, but sometimes that doesn't happen. Sometimes it happens that researchers just think, oh, this is an interesting idea to investigate. So that is one point already to start with a relevant clinical question and the correct clinical question. So then there's a big drop in the potential or the success rate in terms of actually translating it to the clinic. And often you need to have an industry, a commercialization unit to actually work with a company to move it to an actual clinical diagnostic test. So what are some of the difficulties that's in this valley of death, and how can we pass this valley of death? So this figure is from a recent article indicated in the bottom there. Today I will talk about the relevant parameters for proteomic biomarker translation. And some of the boxes, some of the terms I have highlighted here are the reproducibility, clinical relevance, and technical expertise. So that's what was coming from this article. There are several, many other issues that impact on the success of translation, but for today we're going to focus more on the technical proteomics parameters. And for proteomics specifically, I understand in this workshop you have been looking at a lot of different proteomic techniques. I am focusing on mass spectrometry based techniques, but many of these questions and requirements also apply for other techniques as well. So, but particularly for mass spectrometry based proteomics, a lot of the technical focus are very time consuming, require high level of technical expertise. This then poses some issues for analysis of large cohorts. So in terms of the proteomic side, what I wanted to share with you today is the very high importance for reproducibility in the data in generating the mass spectrometry data. This means the methods should be high throughput to enable cohort analysis. This then requires some sort of automation because even for somebody who's very technically skilled in pipetting, if you have to analyze hundreds or even thousands of samples, this will introduce variability in the data. And other parameters that are not often considered is also sample randomization when you run a mass spectrometry batch, and also some quality controls. So when I use our example to illustrate the use of these points, I will point out these particular parameters that I think it's very important to pay attention to these details when designing your workflow and your parameters. So the example I will share with you today is our research project on discovering serum biomarkers for early detection of esophageal adenocarcinoma. So this cancer occurs at the lower part of the esophagus, and it is a very bad prognosis cancer. Most patients are diagnosed at a late stage, and it's then an incurable disease. One advantage, I guess, for this disease is that it has a pre-malignant condition that is known. It is called Barrett's esophagus. So it allows a sort of target screening group to then identify or to try to identify cancers early. And so the pathogenesis of the cancer is illustrated here. So a major risk factor, or how the cancer and the precancer starts, is due to a chronic reflux. So it's also called GERD, gastroesophageal reflux disease. And the prevalence of this, it causes very uncomfortable heartburn, and it's very common in Australia and in Western countries, so affecting 10 to 15% of the adult population. So long-term and chronic GERD can lead to change of the cells that line the esophagus to Barrett's esophagus. So it's a change of the squamous cells that line the esophagus to a columnar cell as a protective mechanism. So this change can occur in up to 2% of the entire population. So it is a fairly common condition. And then if the reflux continues to happen, there's about a 0.1% per year progression through to a low-grade dysplasia and a high-grade dysplasia, and then eventually to esophageal adenocarcinoma. So currently, the survival rates are quite low. 5-year survival about 20%. Median survival is less than 12 months. So in addition to Barrett's esophagus, the risk factors for esophageal adenocarcinoma are fairly well known also. So it's obesity or high body mass index, males, and older age. So how is the cancer and the Barrett's esophagus diagnosed? So the method is used is endoscopy with a biopsy, and this is where the flexible tube is passed down with a camera and a biopsy device. And the gastroenterologist then can look visually and take samples, and it can be then taken to the pathologist and diagnosed by the histopathology. Now, there's a Barrett's esophagus surveillance program that's active in many countries, but it is not very effective in improving the clinical outlook of the patients, because the conversion rate from Barrett's esophagus to adenocarcinoma is very low. And this procedure is quite expensive, so few people, relatively speaking, can be screened in this manner. And also it is quite invasive, and there's a chance of some tissue perforation, and it could be having some different complications. So as a screening method, it is not very successful. And so in the last decade, there has been a lot of research to look for non-endoscopic surveillance methods to catch Barrett's esophagus, or to determine the progression to cancer. And I want to share with you one relatively late stage development that has been developed, led from the UK. So this is a cytosponge device you can see in the picture here. So they identified a transcription factor, which is trefoil factor 3, that is increased in the tissue, in the esophageal tissue. And then they invented this cytosponge device, which is in the capsule, and then given to the patient who swallows it, and then they pull it back up, and thereby sampling the esophageal tissue. And then the tissue is used for immunohistochemistry staining, and you can see some dark stained tissue here as an example. So in this particular paper in Lancet, they did a randomized controlled trial to identify Barrett's esophagus patients using this method and compared to the usual care. And it is a very impressive trial with more than 100 GP clinics and thousands of participants. But this number, well, still quite a large number, 1,600 actually had the cytosponge, and from that, there's 200 positive and had a follow-up endoscopy. So the idea is the cytosponge gives a pre-screen as an indicator to prioritize the patient for endoscopy. And from there, 123, so around 50% who were positive for the TFF3, was diagnosed with Barrett's esophagus, and then 8 were diagnosed with dysplasia or cancer. So how is this performance? Is it good enough? So even though there were more Barrett's esophagus identified to usual care, when they did the comparison, you can see a very low number of the people were identified with dysplasia or cancer. And if we go back to the actual clinical need, so what is the clinical need? What do we want to know? So identifying Barrett's esophagus patients may not be what is required, because at the moment, the treatment stage is high-grade dysplasia. So this is what can be called early cancer, or the actual cancer stage. So when a patient is diagnosed with these 2 stages, they can undergo endoscopic ablation if it's a high-grade dysplasia, or they undergo surgery for the cancer. So really, we want to identify high-grade dysplasia and cancer, not Barrett's esophagus, because Barrett's esophagus patients, they're not treated, it's asymptomatic, they're not really at risk of dying. But why is it that so many research projects have found biomarkers for Barrett's esophagus? So it seems in the pathogenesis, Barrett's esophagus already has genomic and phenotypic changes at this stage, and there's the further changes ongoing to cancer is not as obvious. And so many researchers go with genomics, transcriptomics, methylation analysis, trying to identify early markers for esophageal adenocarcinoma, but they haven't been able to find it. So we applied a proteomics approach and have had success in finding these markers. So when we started, we really considered what is a clinical need, and we discussed with our clinicians quite closely, and they were very keen for a blood test or a liquid biopsy to for screening patients to find early stage esophageal adenocarcinoma. So that means high-grade dysplasia and the cancer. And this is good because it's a non-invasive test and it is cheap. And we had aimed for a multi-marker panel, so we have the sufficient specificity and sensitivity. So blood is easy in the end use. However, for those who know proteomics, you will realize that asking to find a biomarker in the blood is rather challenging for a proteomic research project, because there are a lot of high abundance proteins in the blood, and to do like shotgun proteomics discovery, it involves a lot of fractionation steps or some other methods, which is not very easy to make it high throughput and will be very costly. And so when we try to find markers, we really have to think how to design this project so it will actually have markers that come out the other end and is useful. So in terms of the blood proteomics then, several approaches have been used. So quite old approach is to deplete abundant proteins. More newer approaches include to focus on subproteomes, and this has to be based on the pathobiology. So what we have done in this project I will show you is looking at glycoproteins. Another approach, which is quite relatively new, is to look at extracellular vesicles, also called exosomes or microvesicles. So this we have done also, but today I will show you the glycoproteomics project. So important to really understand the pathophysiology. So when we started, there were already in the literature quite a lot of different studies that report during the disease progression, there are different glycan structures and different glycopeptides which change in the blood and the tissue during the Barrett's esophagus to esophageal adenocarcinoma progression. And so we were fairly confident that we should find something, but again, it's never certain when you're doing a research project. So with this in mind, we also took some inspiration on some existing or already translated tests. So we looked into enrichment of glycoproteins, and we come to look use of lectins to bind different glycoforms. These lectins are different naturally occurring proteins. There are many, many of them, which have been used in glycoscience for decades. Their role in nature and biology is to bind different types of sugars. So seeing that is their normal job, it is a very good strategy to use these type of proteins to capture different glycosylation types and then use it to measure the biomarkers. And we took inspiration from this particular diagnostic test, which was released by Waco Diagnostics already. It's a microfluidic-based clinical immunoanalyzer, which measures a subtype of alpha-fetoprotein, which has a particular ability to bind this lectin called LCA. So we thought, and it's a good strategy to use different lectins. At that stage, most of the studies using lectin to isolate proteins and then conducting proteomics afterwards usually used a mixture of lectins. And so now I go back, and as a reminder, I show you again this valley of death picture. So when we were designing the platform, we wanted to have reproducibility, high throughput and automation, and we want also in the end to have some specificity. So the method we developed then is a semi-automated method which uses a liquid handler for a lot of the workflows. And for the lectins, we don't want to use a combined lectin pool, but rather to have the specificity of individual lectins. So we coupled individual lectins to magnetic beads, and each lectin bead is arrayed into a well of a 96-volt plate. And for internal standard and QC, we actually add in a chicken of albumin to every single sample. And also another consideration which we spend quite a lot of time optimizing is to denature the serum proteins first, because there are a lot of protein aggregates or proteins that bind to other proteins in the serum. And so we consider that if we just use lectin to pull down on the native serum, we might pull down a lot of different protein complexes. And later on, when we do the proteomics, we might be confused in considering that maybe that is not the actual protein target, but it just came down without pull down when it's co-binding. So we spent quite a lot of time optimizing and characterizing and validating our workflow before we actually went to the clinical cohort. But to make it really streamlined, though, after we capture the glycoprotein on these magnetic beads, we don't elute them. We just wash it thoroughly. So I think we do something like 5 different washes and also change the plate twice to remove background. And then we directly do an on-bead digest to release peptides for mass spectrometry. But when we did some immunoblotting for just for cross-checking, then in that case, we elute the proteins. So all the workflow is done on a liquid handler for the processes shown on this slide. The beauty of this technique, though, is at different stages of the biomarker discovery and validation, we use the exact same LEMBA method, but we couple it to shotgun proteomics for the first phase, or to MRM mass spectrometry for the validation of biomarkers. So we have some consistency in this step without changing. So the next consideration for the biomarker pipeline is what kind of samples we should use. and which different cohorts and which different analysis we should use. So I'm just jumping to the end and show you what we did. So even though I mentioned at the start that the biomarkers we want to find are not for late cancer, but actually for early cancer, so called high-grade dysplasia, or HGD, shown in here. For the initial discovery of the biomarkers, though, we just started with very well-characterized samples, which fall into either healthy, as confirmed via endoscopy, Barrett's esophagus, or esophageal adenocarcinoma. And one of the challenges in the clinical research is that because the disease is diagnosed at late stages, there's not too many early cancer samples. So by this way, we can detect and discover candidate biomarkers that can then be taken for evaluation. And so in the first phase, we only had 10 samples per group, and we used a data-dependent acquisition, but we did a screen across 20 different lectins to find the most distinguishing lectins to follow up. And in the second phase, we used a similar design, but increased the sample size and decreased the number of lectins. So we pick up the most likely bias, designed a MRM assay for those candidates, and then use that to discover or to validate the markers in that case. And in the final phase, we reduce the lectins further down to only 4, because each lectin pull down is a separate mass spec run. And so with these different numbers, we are talking about several months of exclusive use of a mass spectrometer. So one needs to really prioritize the different samples. So in the final phase, we have also 2 different cohorts, a relatively large Australian cohort, which we use as a test set, and a clinical cohort from the US, which we used, the Australian cohort was the training set for the model, and the US cohort is a test set. And in this last phase, we also then included high-grade dysplasia samples, which are the real question. So to cut a long story short, these different phases finally led to to a biomarker panel of 10 glycoproteins. So the training cohort had an AUC of 0.93 with 86% sensitivity, 85% specificity. In the test cohort, which was completely independent for the model, it has, as expected, lower sensitivity and specificity. But this statistics, even though not as impressive as the training cohort, is also is quite usable in terms of a screening, because the point of the screening is to reduce and actually identify the right patients to go on for endoscopy. So have we emerged from the valley of death? Well, we are still working on that, but at the moment we are working with Proteomics International to move to a lab-developed test using a LEMBA MRM assay. And they actually have already gone through the valley of death, and they have already commercialized a ProMarker D test, which measures 3 biomarkers that they discovered using the depletion approach. And this again has taken a long time. So for last few minutes, I want to share with you a very recent result to develop the clinical lectin immunoassays. And as I mentioned to you, our original inspiration is this simple equipment that can be used in the doctor's office, who can do a quick test and then say, okay, this patient should be referred to the hospital for endoscopy, or, you know, you are okay. So we actually had been working on this for many, many years, and it's quite challenging to set up new immunoassays. So we started with the top biomarker, which is complement component C9, which we had already validated by tissue immunohistochemistry in our previous paper. And even though it's not important in terms of the diagnostic, just quickly say that C9 is the final molecule of the complement cascade, which is common in the blood, and it is activated by antibodies or proteases or microbes in this proteolytic cascade, and the C9 is the protein that is responsible to punch hole in the target cell, for example, like the invading bacteria. It has been, a complement system has been suggested to be altered in cancer. Sometimes people say it's a response to develop cancer, but we are finding it changed in glycosylation in early cancer, so high-grade dysplasia. So again, to cut a long story short, we had quite a few difficulties developing the lectin immunoassay. In the end, we collaborated with some colleagues at the University of Queensland and developed this endoscreen chip. So it is a microfluidic system with a surface-enhanced Raman spectroscopy detection. And how it works is depicted here. So Jacqueline lectin is immobilized on this chip, and it binds the C9 protein via the glycosylation, which is, it preferentially binds to O-linked galactose on the C9. And then we use a C9 antibody, which has got a SIRS barcode on it to detect. So in this little chip, we first conducted validation of the assay. So we used human serum, which has been depleted of C9, and then we added back the C9 that we had purified. And so we see a good signal in terms of the negative controls, which is the serum, C9 negative serum or PBS. And we see a good signal when we added back C9, either in PBS or in serum. And there's a bit of signal suppression in serum, which is not surprising, but that's why it's important to test it in the right matrix. And then we also confirmed the linearity of the endoscreen chip, which is shown here. And then we went back to one of the cohorts that we had analyzed earlier and checked the changes in the C9 and JAK C9. So the JAK binding C9 is significantly different between B and EAC. And for the first time, actually, we found that it is, for the total serum C9, it is also significantly different between B and EAC. So this had never been reported before. But as a justification for all the trouble we went to for the glycoprotein biomarker discovery, we find that the JAK C9 is a better predictor of EAC with an odds ratio of around 5 for BE versus comparing to EAC, and then also for healthy. Whereas the total C9 is only a very low odds ratio, so less than 1.5. But then the question still is, are our biomarkers very useful? So we did, again, the data modeling to check if it's useful. And so in this case, we used the clinical variables, the risk factors. In the model 1 here, I'm showing the factors of age, body mass index, and reflux history. And that could be used as to predict if the patient has BE or EAC. And this is shown in the red with the AOC of at C9 and the JAK C9 measures that we developed in the immunoassays. And you can see for each of them, we increased the predictive power by about 0.1. So it's looking like the biomarkers could be useful. And so currently what's ongoing with this work is development of the clinical grade lectin NMR assay with the Proteomics International, which will be then followed by clinical studies. And for the lectin immunoassays, we will be then try to multiplex the markers. As I mentioned, we're looking for a panel. And so thank you very much for joining me today. And as a summary for what I've showed you today, the shortcut proteomics is already used in the clinic and making a difference to the patients. And actually for HUPO, I put together a personal story of one of the patients. It's shown on the right here and in the HUPOs. This is available on the web. If you're interested, you can have a read of how it helped a particular patient to really change his life. And in the second part, I shared with you a rather long process for biomarker discovery and translation. So it needs a lot of dedication, a lot of collaboration, but also one needs to be very careful with the study and the techniques that are used to make sure that biomarkers can be discovered and then translated to a useful test. So just to close, I'm just thanking all the people involved in the second study. So thank you for your attention, and I'm happy to take any question.
Yeah. So thank you everyone for listening. This was Michelle's talk. Next we